viral disease goats. As we know that the plethora of organisms affecting livestock, it is very much high and enormously growing, and particularly concerning about the viruses which can bring a various threats to our livestock population. So today, some of the diseases which I'll be discussing about goats and sheep. Let us begin with some salient features of our Indian economy. As per the available estimates of the National Account Statistics, the livestock sector, which is growing significantly at constant prices, and if we look at the gross value added by agriculture and life sector, it has also significantly contributed to the gross value added of to agriculture. And if we talk about the goat population, as per the 20th livestock census, India homes almost around 148.8 million of goats. And out of the many important threats and many constraints which are faced by the farming community which rears goat, it is the disease outstands them. And as per the reviewed literature, presents that the magnitude of such disease leaves a long-lasting and devastating impact to this sector in our country. Next, since I have already mentioned that the list of diseases, it is very enormous and it is still growing. So I'll be considering only 3 diseases in my today's webinar, particularly which are got very high, which are very significant in the sense that these are WOH, World Organization for Animal Health listed diseases. Number 1, the blue tongue disease, Pestides petits ruminants, sheeppox and goatpox. The other listed diseases are Medivisna, which is caused by lentivirus, then Nairobi sheep disease, which is of course a very new disease, caprine arthritis and encephalitis, and one of the most challenging disease to our livestock, which stands out to be great in terms of its magnitude and devastating effects which is caused to the economy, it is the foot and mouth disease virus. Coming next, let us discuss these 3 diseases. First, when we talk about blue tongue, it is an infectious, non-contagious arthropod-borne disease which is caused by a virus belonging to the genus Orbivirus and family Reoviridae. Due to its economic impact, Bluetooth is a World Organization for Animal Health listed disease. And this particular disease, the blue tongue disease, it is a multi-species disease. The economic losses associated with blue tongue virus infection are either directly through morbidity patterns, mortality, poor reproductive and productive performance, or indirectly causing trade restrictions involving animal and animal byproducts. Global losses, as estimated by some of the researchers, were estimated to about 3.3 billion US dollars. The blue tongue virus, it is basically transmitted naturally to susceptible hosts by the bite of blood-sucking midges of the genus Culicoides under the family Ceratopogonidae. There are several other factors which governs the epidemiological cycle of blue tongue virus. It is mentioning a few would be like ambient temperature, humidity, rainfall, and swampy areas favor the distribution, survival, and breeding of Culicoides vectors. Next, when we talk about the virus which is responsible for causing this blue tongue disease, this virus, it is very unique in nature. It is a double-stranded RNA virus which is lacking an envelope, and it has got segments in it like other segmented viruses like orthomyxoviruses. And due to this segmented in nature, this virus, as we can see today, almost there are 28 serotypes of blue tongue virus which has been reported globally, and out of these 28 serotypes, 24 has been recorded so far till date in our country. Moving next, how this disease spreads. Basically, this disease, as I have already mentioned, that it is a non-contagious disease. So when the involvement of the vector culicoides, it is very, very crucial for the transmission of this disease and maintenance of this disease. Now, once there is cutaneous instillation of blue tongue virus by the adult female species of culicoides, when they suck blood as a blood meal, they get up the infection from the infected host, and once they take up the infection, the virus first it multiplies, which is called as extrinsic incubation. The virus first multiplies in an external vector, which is the culicoides, in the midgut, in the cells of the midgut. Then it comes to the salivary gland, and from there, the culicoides, it spreads the disease to other susceptible hosts. And the host, once it acquires the infection, it becomes infective and viremia starts to develop. Once the viremia starts to develop, this infected host, now it can spread the disease to other susceptible hosts, like through culicoides bites, through contact, when they are densely populated, when animals are reared in a densely populated. Particularly this is seen in case of deers, no, which are densely they are kept in captivated, and it is seen in those conditions. Then orally, one, there are certain vices, like when after pasteurization the animals, they ingest their placenta. By that way also they can take up the infection. The other mechanical ways are through various vectors or various arthropods like sheep, cat, and tick. And other unhygienic practices, the virus can be very well transmitted to other susceptible hosts like itros, which we call iatrogenic infections. Another very crucial important aspect of this blue tongue disease is this disease is also spread transplacentally. That means from the pregnant ewes to the lambs. Another way is through natural coitus and through artificial insemination. As we see further, that as we can see further, or I'll be telling in the subsequent slides, that this particular virus, it has also affinity for almost all types of blood cells, and we can see further how this susceptible host can take up the infection. Now, talking about the pathogenesis of this blue tongue virus, this blue tongue virus, it mainly spreads through the bite of infected culicoides species. Once it bites the susceptible host, then the antigen presenting cells, particularly the dendritic cells, it takes up the virus, and then it carries away this virus to the regional lymph nodes where there is primary replication of the virus. And then through the lymphatics, it spreads to the systemic circulation. Once the virus gains access to the systemic circulation, now it is free to move anywhere. So it affects other secondary organs like that of the heart, lungs, uterus, and spleen. In these particular organs, the virus, it has affinity for the mononuclear phagocytic cells and the endothelial cells of various organs. Now, when the virus gets access to these endothelial cells, and when there is leukocyte trafficking of this virus, these endothelial cells, they are severely injured, thereby leading to vascular thrombosis and disseminated intravascular coagulation. Now, once the virus causes endothelial injury, there is release of several cytokines, which particularly promotes, or they are pro-inflammatory in nature. Sometimes in optimal cases, it is very beneficial so that the immune system can take up its work. But in certain responses, when there is aggressive, or when this behavior is very aggressive, that is, there is a condition which is called a cytokine storm. This cytokine storm, it further aggravates the condition. And as I mentioned earlier also, that this particular virus, it has got affinity for all the cells of the blood, particularly the red blood cells, the platelets, all the white blood cells. It has got affinity for these cells, and the virus is trafficked in these particular cells. As I have mentioned earlier, that when there is injury in males, particularly the rams, these the RBCs, they may be found in the semen, and through that also, because these viruses get adhered to these RBCs, and that becomes a very important and potent source of transmission. Next, some of the clinical signs which we can encounter in case of blue tongue, in case of both sheep and goat. As I mentioned, that there are almost 28 serotypes, and these clinical signs and symptoms, it varies according to these serotypes. And some of the serotypes which are very, very pathogenic to sheep, and some may be either more pathogenic to the goats. And some of the common lesions which we can encounter in almost all the serotypes of this blue tongue virus, it is like cyanosis of the tongue, and then particularly nasal discharges from the nostrils, rhinitis. Then there might be edema of the lips. The lips become swollen. So, there's an important term which is called as monkey face. And in particularly young ones, they become very debilitated, reluctant to move. Another important characteristic feature of this blue tongue, in case of small ruminants, it is coronitis, which infects the endothelial cells, and then we can see some of the erythematous lesions in the coronary bands. Particularly in the lungs, we can see diffused interstitial pneumonia, where it's characterized by non-collapsible lungs. The lungs are very edematous. And if we look at the histopathological changes of this particular virus, we can see that it causes injury to the blood vessels of the various organs, and in the lymphoid organs, it causes depletion, severe hemorrhages. Particularly in the brain, also we can see hemorrhages when it affects these small ruminants. Next, as I mentioned, due to this coronitis, there is severe inflammation, particularly the small ruminants. In case of sheep and goat, they become lame. It is very difficult for them to get up their gait. And the other lesions which we can encounter, like swollen face and lips. Then we can see bulbar and palpebral conjunctival hemorrhages, multiple hemorrhages on the conjunctiva of the outer aspect of the third eyelid. The name, as we can see, reddening and extensive erosion and shedding of the mucosa of the dorsal aspects of the tongue. The tongue becomes cyanotic, that is why the name blue tongue. The focal areas of petrification, particularly on the ventral aspect of the tongue. Now, if we talk about the various serotypes which is present globally, as we can see in Africa, it has reported 22 serotypes. Europe has reported 13, North America 16, South America 19, and likewise in Australia 14, East Asia 20 serotypes, South Asia 13. And when we talk about like in the present scenario of blue tongue, in case of Southeast Asian countries, it as of today, it has almost reported 24 serotypes. Now, particularly when we talk about India, Tamil Nadu has reported 18 serotypes, followed by Maharashtra 16, Telangana has reported 17, Tamil Nadu 18, Karnataka 10, Maharashtra 16, and in the other parts of the country like Orissa, West Bengal, they have reported a very few numbers like Uttar Pradesh 4, Uttarakhand 2, Himachal Pradesh 8 serotypes have been reported so far, Jammu and Kashmir 7, Rajasthan has so far reported only 1 serotype. As we review the various works done by the eminent research scholars and research scientists from various nook and corner of our country, we can see that this blue tongue virus, it is very, very prevalent in almost all the states of our country. And then as per the predicted by the National Animal Disease Referral Expert System, it has predicted that in the near future or in the coming days, the states of Gujarat, certain states of this eastern part of our country and South, they are very, very prone to suffer from this blue tongue virus. Now, to curb this menace, in India, we can find a vaccine which includes polyvalent serotypes like 1, 2, 10, 16, and 23, which is containing inactivated vaccine of the blue tongue virus. And it has been manufactured and marketed by the Indian Immunologicals as the name Raksha Blue. Another vaccine which is available in the ICR Indian Veterinary Research Institute. It is also a uh, it was developed under All India Network Project. It is also a first multivalent inactivated vaccine which contains the serotype 1 to 10, 16, and 23. And more or less, the vaccination schedule is same for both the vaccines. The another very important disease which affects particularly the goats or the small ruminants, it is the Pastides petits ruminants. Otherwise, the synonym for this particular disease, it is goat plague. It is a very highly acute contagious disease and devastating disease of small ruminants. Other synonyms: erosive stomatitis, enteritis of goats, pseudo rinderpest, pneumoenteritis complex, This disease, it is commonly called as goat plague. Now, earlier, the virus or the etiology for this particular disease, it has been renomenclated, or the new name for this particular disease is morbillivirus caprine virus. And the epidemiology of pestis death pestis ruminants, it was first recognized in 1942 in the Ivory Coast of West Africa, and in India it was first reported from a district, Villupuram district, and the mortality rates is as high as 50%, and it can also, in some outbreaks, it can also reach up to 90%. Now, when we talk about the epidemiology of this PPR virus, apart from the small ruminants, the virus is also present in other wild ruminants, and it has been also reported from buffaloes, cattle, and camels. In some of the species, like when we talk about pigs, they are reported to be infected by this virus, but they are considered to be as dead and host. Cattle are usually infected asymptomatically and are not known to transmit the disease to other animals so far. It is caused by paramyxoviridae morbillivirus, which is a single-stranded RNA virus enclosed in a ribonucleoprotein core. Now the nascent nomenclature, it is morbillivirus caprine virus. And in this particular virus, we can see intranuclear and both the intranuclear and intracytoplasmic inclusion bodies in the infected cells. Transmission, as I said, it is a very contagious disease. So it is from infected animal. The most probable or the most frequent route of transmission, it is through inhalation. And the virus, it is excreted from various secretions and excretions like nasal and ocular secretions, saliva, urine, and feces. The pathogenesis of this particular virus, it enters through the host mostly through inhalation. The virus, it is initially taken up by the antigen presenting cells. Then it goes to the nearby regional lymph nodes, transported to the lymphoid tissues where primary replication takes place. The virus particularly has affinity for the lymphocytes and infects throughout the body via lymphatics and the vascular system. This particular virus, the PPR virus, it is known to have affinity for the lymphocytes as well as the epithelial cells. And then thereby how it causes the pathology, it causes conjunctivitis, rhinotracheitis, ulcerative stomatitis, gastroenteritis, and severe pneumonia. There is also, you can also see in the lymphoid organs, there will be extensive necrosis in the Peyer's patches, spleen, and in the pulmonary lymph nodes. Now, since it has got affinity for these leukocytes are destroyed, thereby causing severe immunosuppression. And once the animals becomes severely immunocompromised, they become very prone to other concurrent infections like that of pneumonia and other protozoal infections. This particular disease, Pestides pestis ruminans, it is clinically manifested in various forms, like acute form. It is mainly seen in goats. Signs appear within 3 to 6 days. The most characteristic signs of this acute form are fever, dullness, sneezing, severe nasal discharge from the ocular discharges and nasal discharges. Then there is severe leukopenia. Once the oral cavity is affected, there is severe foul smelling from the infected animal, halitosis, and diarrhea is also very frequent in case of PPR. Deaths usually occurs within a week when the case is acute. When the onset of this disease is a very acute, death is a final outcome, and it is very frequent in case of acute form. In subacute form, which is mostly recognized in sheep, it is recognized by low-grade fever, and the diarrhea, it is intermittent. Mostly it is when these animals are subacutely infected, they recover in the due course of time. Animals after recovering have a lasting immunity. The gross lesions like discharges from the eyes and nose in advanced infections, reddening of the conjunctiva and purulent eye discharges, gray areas of dead cells on the gums. Sometimes you may also notice brain-like depositions and severe erosions on the mouth of the affected ruminants, small ruminants, which is responsible for causing of this halitosis or foul smelling. The hindquarters of the affected animals are soiled with semi-solid feces. and you can also encounter nodular type of regions around the mouth. One of the characteristic feature of PPR, it is bronchointerstitial pneumonia, particularly in the ventral areas of the lung, areas of consolidation and atelectasis. Another pathognomonic lesion for this PPR, it is in the ileocecal of the region of the rectum, where we can notice zebra stripes or zebra marking. Microscopically, in this infections by this virus, we can see in infected tissues, we can see multinucleated giant cells, particularly in the epithelial cells of the upper respiratory tract. And in this particular infection, which is very characteristic for PPR, we can see both inclusion bodies, that is the intracytoplasmic and the intranuclear inclusion bodies. When it affects the lungs, we can see severe proliferation of the type 2 pneumocytes and formation of syncytial giant cells. In the lungs, we can see that the interlobular septa, it is thickened by the accumulation of fibrin and cellular infiltrates. The alveoli, they are massively infiltrated. Here in the alveoli, there is massive infiltration of inflammatory cells. And diagnosis, it can be done like differentially. It can be diagnosed with complement fixation and virus neutralization test. Confirmatory diagnosis, we need to take up advanced techniques like reverse transcriptase PCR, differentiating it from PPR or from RP. Now, postmortem lesions, which is now in this picture, which is showing the congestion of lungs and lungs lobe. Lungs lobes are edematous, they are non-collapsible, and the lymph nodes are very, very congested and enlarged. The colon here, it is showing hemorrhagic, linear hemorrhagic streaks, and we can also encounter like erosive lesions in the mouth. These are other lesions which we can encounter in this particular PPR, erosions in the mouth, consolidation of the lungs, and when these consolidated lungs are incised, we can see fatty accumulation in the trachea. Then we can find intracytoplasmic inclusion bodies in the affected epithelial cells. And to tackle up this disease, the most important preventive measure is to go for vaccination. Vaccine, it is available, marketed by the some of the renowned vaccine manufacturing companies like Heister, Indian Immunological, and the age primary vaccination, it is for 4 months and above, and it is again revaccinated after every 3 years. Another important disease which is also notifiable to the World Organization for Animal Health, it is a pox disease caused by pox virus. It is a DNA virus, contains single linear molecule of double-stranded DNA, and these multiply and mature in the cytoplasm of host cells. The sites of multiplication are visible through light microscopy. We can see them in light microscope. Now, when we talk about the pathogenesis of pox in the skin, the virus spreads from the migrating lymph nodes. They act as trafficking carriers for these particular viruses. There is direct injury to the vasculature and an acute inflammatory response develops, whereby leading to active hyperemia and active inflammation. And then we can see the several stages of pox, macules or development of macules or rhodiola stage. Then the virus from the capillary endothelial cells and trafficking leukocytes infect the Langerhans cells, which then the virus again it is spread to the contiguous skin epithelial cells. The proliferation of epithelial cells of particularly the stratum basale and spinosum. There we can see the papular stage, epithelial cells undergoes ballooning degeneration, and the serous exudates infiltrate and detach the degenerated cell. And finally, the vesicular stage. The leukocytes enter into these vesicles, then they take up this inflammatory process, they particularly digest the debris, and then pustular stage is formed. And finally, this pustule, when it erupts, it is covered by a craft or it is covered by a crust, this squamative stage. The other diseases which is caused by capripox, it is sheeppox and goatpox, and other very important rampant disease which is now very wild in our country, that is lump skin disease. Now, particularly talking about the sheep pox, it is a very highly devastating systemic viral disease of sheep. It is characterized by fever, generalized papules. It is most serious among the pox disease. It causes extensive economic loss through mortality and reduction in the productive traits of the infected animal. And it also draws a huge amount of financial burden in order to prevent this particular disease. And the sources of this sheeppox virus, it is through ulcerated papules on the mucous membranes of infected animals, the skin lesions of the infected animals, and the virus is also very well secreted in the saliva, nasal, and other secretions. The virus has also been reported to be present in milk, urine, and feces. And so far, whether this virus it is venerally transmitted or not, it is not yet established. The pathogenesis of both the viruses, the sheeppox and the goatpox, I'll be taking it together. The virus enters mostly through inhalation or ingestion. It is firstly deposited in the mucosa of the oronasal pharynx, especially in the tonsils. Then the virus infects and replicates in the epithelial cells, lymphocytes, and macrophages. The macrophage, when the virus affects these macrophages, they spread them into the regional lymph nodes. In the lymph nodes, pro-inflammatory cytokines and chemokines are released from the infected macrophages. They recruit naive lymphocytes and which are infected with virus. That is how there is trafficking of this virus. Virus then spreads circulatory system via leukocyte trafficking and also through the lymphatics. Then secondarily, the virus, once it gain access to the systemic circulation, it spreads to the other important organs like lymph node, spleen, and bone marrow. It infects and replicates in similar cells using mechanisms which I have already described earlier. The virus then spreads from systemic lymphoid tissue to the skin, lung, liver, and other organs. In those organs, the virus produces the lesions which I have described earlier. The clinical picture of sheeppox, it particularly varies. Like there are many factors which is responsible for variations of the clinical side. It depends on breed, age, the status of the infected animals. The lesions undergo same macular, papular, vesicular, and pustular stages typical to that of a pox disease. Some animals develop numerous lesions and become severely ill. The others, they mild develop some certain mild symptoms or certain mild symptoms with no clinical signs. It varies. Once the virus infects the cells, particularly the cells of the eyelids, it leads to blepharitis. Thereby, the eyelids get pasted. Then there is severe mucopurulent discharges, and we can also see some necrotic patches, particularly in the hairless regions of the prepuce and vagina, very necrotic. In certain female animals, the perivaginal area and the perianal area, we can see lesions of pox. There is severe noisy bleeding due to this in enlargement of the lymph nodes, particularly when the bronchial lymph nodes in the lungs are affected. They cause severe respiratory distress in the infected animals. And the lesion starts to develop with like small nodules, and then finally it spreads to other. Particularly, it can be seen in the hairless body parts, like mostly in the axillary regions. These are the nodular regions which we can see in the infected sheep. As I have mentioned that the nodules are of varying size. It is not uniform in size. It may vary. The diameter is varying in case of infected animals. The other pictures, these are some of the lesions which we can encounter in case of sheeppox. Then when the case becomes very severe, there will be severe nasal discharges and rhinitis. There will be mucopurulent discharges from the infected animals. Now, mostly these particular lesions are seen in the skin. Apart from these skin lesions, the virus can also infect the other organs like lymph nodes, lungs, and other important organs. This is infected, lung is infected with pox virus, where we can see various nodules of ununiform sizes, particularly diffusely spread in the lungs. In case of internal organs, we can also encounter certain nodular lesions in the intestines of the infected animal. And in the histopathology or the microscopic lesion which we can encounter in sheeppox, one of the characteristic feature it is the formation of eosinophilic intracytoplasmic inclusion bodies in the infected cells, and particularly we can see erythematous lesions characterized by marked edema and hyperemia, and there will be infiltration of inflammatory cells. In certain cases, we can also see severe necrotizing vasculitis noticed in arterioles and post-capillary venules. On the lungs contain intracytoplasmic inclusion bodies. Additional histologic lesions characterized by the accumulation of sheeppox cells may involve heart, kidney, liver, and other organs may also be affected with this particular virus. And these are the characteristic inclusion bodies which is seen in case of infected cells infected by pox virus. The goat pox, it is also similar to that of sheep pox, and both are possibly immunologically related. The disease has somewhat longer incubation period than that of the sheep pox, and as reviewed from the literature, this goat pox, it is less severe than that of sheep pox. Clinically, this particular goat pox, it is very, very similar to that of sheep pox, and young kids suffer a systemic disease with lesions generally spread over the skin and other important organs, like the respiratory tract and alimentary mucosa. These are some of the lesions which we can encounter in goat pox, where we can see nodules, particularly in the areas where it is less here, in the groin areas and in the perianal areas, and also we can see throughout the skin when there is very severe infection. These are some of the gross images which we can see some of the lesions in the affected organs. Sometimes when it is very severe, we can also see eruptions in the tongue, and we can see that the organs are severely congested, and we can also see pericardial hemorrhages in case of infected when it multiplies in the cardiac myocytes. These are some of the lesions which I have collected from various sources, which are not from various sources I have collected to just show, and these are confirmed to be that of pox. Now, there are vaccines available, which is produced by the Indian Veterinary Research Institute, and also there are certain other commercial manufacturers of vaccines for goat pox and sheep pox, and this disease can be very well controlled by going for vaccination using vaccines which are prepared by renowned vaccine producing and marketers.